All right, it's 5:02. That's just three people. Let's give it a couple more minutes and then I'll start the class. So I'll start in 3 minutes. All right, so 5:05, I'm gonna begin. All right, so you should be seeing my screen at the moment. Um, I think I'd indicated I had sent the this PowerPoint to Alea and asked her to forward it to you guys. But it occurs to me that I actually have a Canvas account, right? And I could actually put it on Canvas. I don't know if you guys know what Canvas is. It's a program that's very similar to Schoology, which I'm sure many of you probably got somewhat used to. And on Canvas, I actually have a genetics course here, as well as the anatomy and physiology one already. That is an AMP1 course. So what I'm thinking about doing is sending you all the registration information for the genetics course so that you can log into Canvas and you can access the PowerPoint presentations, etc., from Canvas, right? But let me ask, do any of you have a Canvas account? Anybody has a Canvas account?
No, no, sir.
All right. Um, it's pretty straightforward. It's pretty easy to register an account for. So what I'll do is, well, all right, give me one quick second here, and let me see if I can just send you the login info for Canvas. Um, at least that way we have it in case anything else, right? So just give me like 2 minutes. I want to stop the share for real quick and then send you the info for Canva. All right. I'm about to post something in the chat, which is a link to this Canvas. I guess you could call it the online course. That's basically what it is, right? So just go to the chat after I post this and tell me if any of you can access this. Right? So there should be a link in here to register. Well, there should be a registration code, which is 46CE8Y, I think it is. And there should also be a registration URL there as well, okay? And I will sort out something for the genetics as well. But at least you will be able to get the information on Mendelian genetics and stuff like that, so we can start with that, right? We'll figure out the rest of it. Oh, why wait on mood, right? We'll figure mood out in a little bit. But in the meantime, we can at least work with that. All right. Y'all see the link? Any issues there so far? Okay. Now this current PowerPoint presentation is not on Canvas, but there are other genetics links there. I will put this PowerPoint up on Canvas after class, as well as some other information as required. So we're going to try and work with this until we figure out what's going on with Moodle. It might actually be a case where we might not be getting Moodle this semester, I'm told, because technically speaking, the summer semester is, as I said, is not a normal semester. And because it's not a normal semester, some of the rights and privileges, let's call it, that you're used to having during a normal semester might not translate, okay? So please keep that in mind. I'm still gonna try, but I may not succeed, right? Okay. So now please remember, try to get onto Canvas using the URL and the password I have down there at the bottom of the screen in the chat. And please let me know if it does not work, okay? And tomorrow in the AMP class, I'll also share one with you for the AMP2 as well, and we'll see if we can get that going, right? So at the very least, we can probably try to some practice online quizzes, stuff like that on Canvas, keeping in mind, well, I'm going to ask about the potential to do at least one online in-course test if we can get Canvas running properly. Because I do know we have at least one or two students that are not potentially physically nearby, as in that may not in the parish, but they may not be on the island at all, which of course is one of the reasons why they were doing things online to assist, right? So I'm gonna need you guys to just go and make sure you have your Canvas working based on what I've sent you, and then we can see how we can work things from there. Okay? So as soon as I can, just make sure for the Canvas submit, please, right?
Mine
All
is
right.
working, sir.
Okay, cool. Somebody said it's working. Awesome. So at least one person has it. So all right. You know what? Let me just add this PowerPoint presentation I have here. And then at least we can say, all right, we're in good stead. So I'm gonna add something to this here. Just give me 1 second and let me do it right now before I forget. All right, there we go. So I've just added it. It'll be the last thing. Well, let me see if I can move it to the top. Awesome. So here we go. Mendelian genetics, top of the page. So the PowerPoint we're going through now, you should have direct access to it now as we speak, right? So whenever you get the chance, download it. Anyway, let's continue from where we left off last week. We didn't do a whole lot last week because, of course, last week was basically our beginning week, right? I think it was, or close enough to that. So we did Mendel and the whole piece thing already, and we spoke about genetics. And we spoke about some of the terminology that you need to know already. Spoke about the law of segregation and independent assortment. Just to remind you, segregation, as it says here, parents contribute only one of its alleles for a trait to each offspring, right? So basically, this one is indicating that parents have, of course, two alleles for each trait. Only one of the two will be passed on to an offspring. The one that is passed on to the offspring will be random, right? Of course, that leads to this independent assortment business. So the alleles passed on of each pair to an offspring is random. So it is independent of the others. Okay? So there's one more time just to make sure. Segregation basically means two alleles per parent They're separated, so only one allele goes into each gamete. Okay, that's segregation. Independent assortment, any of the gametes that are, sorry, any of the alleles that are put into a gamete is randomly placed, right? So if you have 21 pairs of alleles, sorry, pairs of chromosomes, then you know only one of each chromosome will be put into a sex cell. All right. So one of each pair. So the combination of the chromosomes that are placed in the cell will, of course, have one of each of your chromosomes, but you can never tell which one of the two pairs for each one will end up in your chromosome. That is what causes the independent assortment. You'll never be 100% certain about the combination of alleles that will be found in a gamete because it's very random. Okay? And please remember why randomness is important. Sexual reproduction requires random chance, right? It requires mixing up of genes, chromosomes, etc., to allow potentially for new things to pop up out of nowhere, right? Now, this here is showing two genes. This is a fruit fly, right? I think this is chromatogast, no, melanogasta, right? So here we have fruit fly with red eyes and fruit flies with white eyes. And the idea here is that the genes for red eyes and white eyes, of course, are independent of one another. If you look at the abdomen, you can see the abdomen for this one looks curved, while the abdomen for this one looks well more normal. So that is also another random trait, right? You can never tell which kind of combination of genes within your chromosome, within your gametes, will lead to this, right? So as it says here, now alleles of one gene are passed to offspring independent of alleles of the other gene. So therefore, if you have parents like this, red eyes, round abdomen, right? And they have another one that has white eyes and like a curved abdomen. When they have offspring, you can have offspring that have white eyes and normal abdomens, or red eyes and curved abdomen, based simply on the fact that the genes for both have a potential chance of being passed on to the offspring, right? And you never can tell which offspring will carry what trait because of independent assortment, right? So because of that, we can say, hey, guess what? At your creation, right? Your was a one. I think I'd read a number that the chance of someone having a child, your parents, of course, having a child that is genetically identical to you is 1 in 13 trillion. There have never been a trillion people on this planet. So statistically speaking, that means that the probability of another you being born is pretty much nil, right? So from that perspective, you are a unique combination of genetics, right? And sexual reproduction cannot produce another you. It might be able to produce genetically people who are genetically very close to you, right? But never your unique combination of genes, right? All right. So this here now is a dihybrid cross, right?
Sir, quick question.
Yes.
What about twins?
What about who?
Twins, sir.
Did you say cloning?
Twins.
Twins. Okay. Twins are a different set of circumstances. Let me explain why. Well, first of all, there are two types of twins, as I'm sure you know. You have fraternal twins and you have maternal twins, okay? Maternal twins are the ones we normally refer to when we say twins. Maternal twins basically are coming from the same genetic stock. By that I mean egg and sperm meet, form the zygote, well, form the embryo, and then cells from the embryo split off, which means that they're both coming from the already fertilized egg, right? And therefore they carry the exact same genes. They're basically, fundamentally they're clones of each other, right? But the chance of an egg and a sperm meeting with the genetics in there that are perfectly matched to merge together and form another you is not going to happen. 13 trillion is a very big number, right? It's just not going to happen. I mean, yes, we can say, oh, sometimes people are born. Yeah, I don't know if you've ever heard of this term, doppelganger. So it's a German term. The idea is that everybody on the planet has somebody out there that is their exact duplicate. Or when I was growing up, the idea was of the evil twin. You know? So somebody out there looks exactly like you, but it's just the evil version of you. I mean, technically speaking, you could be the evil version and they could be the good version, but either way, right? That's pretty much impossible. Yes, it's possible that you can come across somebody and somebody say, boy, the two of you look exactly alike. You never do look exactly alike. All that is happening is that people are concentrating on the similarities between you and some other person and ignoring the differences, right? And even if you looked exactly alike, same height, same physical features, I can say to you that every mole on your body is not going to be the same. When you do a DNA test, unless you're an actual maternal twin, you're not going to be the same. And even maternal twins, as you get older, your DNA shifts a little away from each other. So even maternal twins are not 100% identical as they get older, right? Whereas fraternal twins, of course, are two different sperm, two different egg. It just happened that the mother produced two eggs at the same time, and they both got fertilized within the same time frame. So genetically, you and that twin are significantly different, right? Okay. So now let's talk about the dihybrid cross. We did monohybrid cross last week, where we looked at one trait and then showed how we do a Punnett square to explain how that trait could potentially be passed on in the offspring, right? This time around, we're going to do a dihybrid cross. Now, a dihybrid cross, I mean, obviously, you know, mono means one and di means two. So your dihybrid cross now, you're looking at two traits to be used versus one trait to be used when you're doing the cross, right? So just like in the monohybrid cross, you're gonna have to represent both traits in your Punnett square, okay? So for example, they're showing here, where are these look like fruit flies, okay? So we have fertilization of which parent differ, right? So here we have what we call the wild type here. And by the way, normally when they say something is the wild type, they mean that's the normal type you'd find. That's the usual type, right? Or that's the original type. So for example, if I wanted to, I could say, hey, guess what? Human beings, wild type original was African. So the Africans would have been our wild type. And every other sub, well, we can't even say subspecies because human beings don't have subspecies. We're all the same species. You can say subclass if you want, but even that sounds a little weird because normally when you say sub, you mean below. So we don't usually use these terms with reference to humans. But technically speaking, yeah, we do have subclasses, right? Anyway, but remember, sub doesn't necessarily mean worse from this perspective. It simply means other than the original, okay? So a smaller category than the original. Just keep that in mind, right? Anyway, so we have here our wild type, which of course, as we say, is going to be our original class. And we have two alternates to that. We have this black fruit fly with normal wings. And we have this I guess it's a normal body brown and gray fruit fly with vestigial wings or short wings. And you see their eyes are the same color. They have the same number of legs. They have the same number of body segments. Everything else is pretty much the same, right? Now, when you cross now, these, let's say you're crossing, well, here we have let's say they're using V, capital V here, and capital E. So if you're looking carefully now, you can see that V is going to stand for the wings, right? So when you have capital V, you have normal wings, as you can see here. Two common V's, of course, you have vestigial wings. So that means, of course, that capital V is dominant, common V is recessive, because you need two common V's to get the vestigial wings. For the body, the normal body color, which is this banded color here, is normal, right? You can see capital E here. I wonder why they put E for this, but whatever. But if you want the black body, for them now it's two E's, two small E's, which means, of course, that the ebony color is recessive, right? Now, keep that in mind and then look at the parents that they've chosen. We have one parent that is capital V, common V, capital E, common E, okay? So this parent now is heterozygous for these two traits, okay? Heterozygous for these two traits. So remember when we crossed heterozygous in the monohybrid cross? Remember I was saying to you, anytime you cross heterozygous under normal conditions with a monohybrid cross, you're expected to get a ratio of 3:1 phenotypically, right? So phenotypic ratio, 3:1. Because remember, we got 4 offspring from the cross. Now, obviously, we're not going to getting 4 offspring from our dihybrid cross. You can see how many offspring we get here, right? All right. What I'm just gonna run the dihybrid cross, and you'll see how I come back. But it's pretty straightforward, right? Tell you what, to make life easier, I think I'm just gonna run it in Excel. Just so I can get my 4x4 easier. All right. Okay, so here is my little grid. All right. Now remember how this goes. Let's copy this real quick, and we'll plop it down in PowerPoint, and then we can start. This should work. This is a picture. All right, cool. Now, obviously, I'm not going to use all of this, right? But I should still be able to get my sections, right? So when you look here, we have 1, 2, 3, 4 across, 1, 2, 3, 4 down with our gametes on the outside, right? So we need 1, 2, 3, 4, 1, 2, 3, 4. So we should be good here. So I put the gametes on the outside here, right? Now it says here the first parent So that's the P generation, is capital V common V, capital E common E. So they are heterozygous. And the next parent is also heterozygous, right? So remember now, we're starting. Let me see if I can write on this. Remember now, our parents are like this. Right? So both parents are heterozygous. Now, of course, we need to determine what are the potential gametes that you can be passing on to the next generation. Now, when you're doing the gametes for heterozygous crosses, you have to ensure that there is one of each gamete, one of each other traits in the gamete, right? So if we look here, we have capital V, common V. So our first trait, of course, is V. Then we have capital E, common E. So our next trait, of course, is E. So what we need now are all possible combinations that have at least one, well, and have one V and one E in them to represent a gamete, okay? So when we're doing the gametes, we go back now and we say, okay, of the V, we have two alternatives to V. We have capital V, common V, right? So that, of course, means we must have a gamete that has capital V in there and gametes that have common V in them, okay? Yeah, it's hard to tell the difference between capital and common V. Let me make it a little smaller. All right, we'll do that. All right, so that's common V. So we know, so based on what we have here, we need, our gametes must contain capital V or common V. They must also contain capital E or common E and any possible combinations, right? So clearly we can have capital V, capital E for one gamete, all right? But we can also have a combination of capital V and capital E here as well, right? So this is two types of E. So we can put that down there. Now, there are no more combinations that would have capital V in there that would give me anything new. So I'll take these two for capital V. And then now we have to look and say, okay, what can we do with common V? So we can have common V and capital E, that is over there. And we can have common V, common E, all right? Now I have to look and say, boy, are there any other combinations of V and E that I can get except for these? So remember, we have capital V, common E, capital V, sorry, capital V, capital E, capital V, common E. We have common V, capital E, and the common V, common E, okay? So these are the potential combinations of the two traits that you can find in the gametes, right? All right, so that is how they would have come by this here. So you see what they have here? They have capital V, capital E, capital V, common E, and of course, common V, capital E, and common V, common E. So that is the process they would have used to come by them, okay? So I know you all would have done this in high school, but sometimes this is hit or miss in high school. Any questions here, or we're all good? All right. So if I give you something else that doesn't have these in there, but still have the same basic idea, you should be able to figure out the combination of gametes, right? Any questions where that's concerned? Are you all comfortable with this?
Yes, sir, comfortable.
All
Yes,
right.
sir.
All right, awesome. Just to make sure, let's do one real quick. So let's use Let's say we're doing a plant. So we have T. And then we have let's see, capital P for purple flowers. You get a lot of this from Mendel, right? And then we say common T, short plant, and common P, so you have a little stalk on top of the common, equals white. Remember I said to you that generally speaking, when it comes to color, white is the default. Unless nature is trying to do something, it usually ends up looking white when something goes wrong, okay? Now, let's say I have two parents. I have one like this. And the other one is like this. So those are my two parents. Remember, when you're writing the gametes, you always put the capital one first because it is the dominant one, right? So the dominant allele is written as a capital. It comes first. So can you all tell me now, what are the combinations I can get from the first parent here? Combination of gametes that can come from the first one. All right, 10 minutes easy. I'm just going to ask each person to give me one. And that way I know everybody is good. Shante, can you give me a combination here, please? Shante Douglas, are you there?
Yes, sir.
Yes.
Um,
So let's give me any combination you can from the first parent.
capital T, common P.
Capital T, common P makes perfect sense. Lovely. Jody, can I get one too? From the same first parent.
Common T, common P.
Exactly. Wonderful. Tashania, what about the second parent? Can you give me one?
Yes, sir. Common T, common P.
That works. And then Jadan, what do you think?
Capital.
But remember, for the
Common
second
T,
parent.
capital P.
Come again?
Common T, capital P.
Common T, capital P. Exactly. So we're all on the same page. Wonderful. Now, that said, we can go back to our Punnett square here. We now know our combinations of genes for our gametes. Both parents have the exact same combination, right? So we know what to do next. So we're gonna, of course, fill out our Punnett square. Remember, normally when you do it like this, you don't use that section there. Instead, you use the top row and the first column, right? So we can put in our thing like that. Generating big and obvious that they're common, right? And then, of course, for that same thing over here, right? Right. So once we have our gametes down, of course, it works just the same way it did when we were doing the monohybrid cross. We start on our little box. We look to the left. We look up, look on top. Whatever we have there is what we put. Now remember, we always have to put our common gametes, our common genes together. So the V's go together, E's go together, right? The same letters always go together. So we have two capital V there and two capital E. So that's fine. Then we have two capital V's again. Now we have a capital E and a common E. And of course, the capital E has to come first. Because remember what we said, capitals are dominant, therefore they come first, right? Then we have a capital and a common V. Once again, capital goes first. And we have two common E, capital E's. All right, so I'm gonna fill this out. But I think what I'm gonna do is I'm gonna toss in a few errors in here. And I want you all to look at it when I'm done and tell me where my errors are, okay? So let me just run through this now. Let's pretend that's a E. All right. So I'm just about done. Okay. So what I want you to do for me now is we'll run through row by row. So I'm gonna go ahead with my little highlighter. And you're going to indicate for me as I'm going along, 1, 2, 3, which ones are incorrect, okay? Just to make sure that we're on the same page, right? So I'm going row by row. So I'll start here. So you just let me know when you see something wrong. Just let me know, okay? Just the wrong one. So we have here, then we have here, then we have this one, and then we have this one here. So
Wrong,
all is well for row 1?
No, sir, that one wrong, sir.
Which
The
one?
one that you have the highlight over.
The last one.
What's wrong with this one?
Common
Sir,
V.
you have two capital.
Okay, so we have that there should be a common V. All right, good. So we made our change. All right, let me change the color. All right, then here's our next one now. So here's 1, there's 2, there's Mr. 3, then 4.
Sir, that's wrong, sir. We just
Three.
What, 3 or 4?
Three.
3?
3.
What happened to 3?
Capital V, common V. Capital E, common E.
So what do you want me to change?
This is what you should have there. Capital
Oh, that's
V.
what I should have. Okay, so you're saying I should have a capital V first, then the common
Yes,
V. Okay, yes,
common
I
V.
agree.
Yes, sir.
So we throw that out. And then now we should have, as you rightly said, a capital V and a common V. All right. Are we good otherwise? For that row?
No, sir. The last one wrong again, sir.
What's wrong with the last one? What's wrong with the last one? We have capital V, common V, and we have common E, common E. So what's wrong with
V
it?
should co
Hmm?
V should come first, sir.
Yes. But here's the reason the V should come first. I agree with you. But the V comes first because that's what you started with. Okay? If I decided to start with E's first, then there would not really have been anything wrong with that. but I would have to have been consistent. Okay, just keep in mind, whatever you start with, that's what you must always do. So since I started with V's, everybody else must also have V's first. Okay? All right. Third line now. We have the first one here. Then the second one. Then the third one here. Then the fourth one in this line. All is well here? Is all well for this one? So this one's good?
The
All
second
right.
one, they have two capital V.
Which one?
The second one.
Second one has capital V,
This
capital
one.
V, capital E, comma E. So
Okay,
that
so
one's
it should
good?
be capital V, common V.
Capital V, common V, that makes sense. So that V there should be common. All right. All right, nice. All right, anything else in this line? Okay, we do the last line. Let's see. All right, no objections. So in the last line, no. We have this here. Then we have this. Then that one here. And then number 4. So the last line is okay?
The first one, sir, the dominant V should go first, which is the
All right, so
capital
the
V.
first one here, we swap the V's. Okay. That makes sense. Otherwise, we're good? Oh, what am I doing? The foolishness. Yeah. So otherwise, are we good?
Capital V, common V for the second one as well.
Second one is capital V, common V. Mm-hmm. All right. Okay, so we're good now. Yeah, we'll move on to the next thing.
The third
Sir,
one, sir.
Mm-hmm.
the third one.
What happened to it?
All right, so common V, capital E, and then common E.
Exactly. So so we have our V's first because that's what we started at. But E, well, we should still always have the capitals first in the pairs. So this one should be capital E, common E. Excellent. Now I have great confidence that you all can do this as well. So that's good. So that, of course, is how we come by this, okay? So this should be the same thing that we have here. Now, that said, this one here now shows the phenotype, okay? So this chart here shows the genotype, and this one down here, so now, shows the phenotype. And y'all should remember the difference between genotype and phenotype, right? Genotype refers to the actual genes that are present in the organism, whereas phenotype looks at the
Physical.
physical what though?
Physical characteristics like height and so on, eye color, and those things.
Right. But the phenotype, or as you started to say, the physical characteristics are usually an expression of the gene. With, of course, the caveat that environment affects the way the gene, that the phenotype is expressed, right? Remember I was saying to you last week about height, where you could have the genes to be 7 foot tall, but if you don't get proper nutrition when you're young, it's just not gonna happen. right? So the genotype directly impacts the way the phenotype is expressed, but the environment also does so, okay? By the way, there's an interesting study that was done a few years back, right? See if you think this is a reasonable thing. The more intellectual work you give a child, right? So quote unquote harder work, the greater their problem-solving ability tends to be later on. Does that make sense? Would you agree with that?
Repeat that, sir. I didn't hear you.
The more intellectually taxing work you give a child when they're younger, right? The better their problem-solving capabilities will tend to be as adults.
That makes sense.
That makes sense. So the idea here is that when you have children, you must pressure them. You must give them the hard work to do. Give them difficult problems to solve. Because it's just that stage where they're, in this case here, the genotype, which is their intellectual capacity for problem solving, if you want to think of it that way, must be trained in order for it to express itself in the phenotype. So that's why we're having some problems these days with the younger ones, little ones coming up. They have a serious inability to solve intellectual problems because them get too much multiple choice things and stuff like that. That is why now PEP, I don't know if you all know this, but PEP is murdered little bit in them now. Because PEP is filled now with lots of intellectually challenging questions. And it is causing a real issue. Right? Kids are failing like crazy. Right? Because once again, your genotype, right, is affected by the expression, the phenotype, which is a problem solving ability, is affected by your environment. If you have the greatest problem solving in the world, but you never use it, then you tend to lose it. Sad to say, but true, right? I think somebody had a question. I don't know if they still do.
Yes, sir. Sir, so let's say you have a tall gene and um you're short because of environmental factors.
Right.
Let's say you don't have the right nutrition. Is it that you now have the short gene?
No. You still have
Mhm.
the genes for height. But remember that when it comes to growth, when we were talking about bones, we said, boy, up until the age of 25, you're growing, right? But once you go beyond that, there's no more gain for you. So if you had very, very poor nutrition in the first, let's say, 15 years of life, because this is when the bulk of the growth is going to happen. Even if you just start to get excellent nutrition for the last few years of your growth period, you're still going to be short, right? But if you have children and these children are properly fed and they inherit your genes for height, they're going to be tall, right? That's why sometimes you'll see like really short people with like very, very tall children. You're like, that's crazy. How is that possible? When you look into it, more often than not, you'll find that the diets of one or both of their parents were not very good when they were young. Right? And because of that, it
Sir.
affected the way they looked. Obviously some people can go through poverty and all kind of things and cause that to happen. But also, if you think about it, parents that are not serious about their children's nutrition will also have malnourished children because children love sweets, right? But children need proteins, right? And fats, not just carbohydrates in order to grow. So if you have a child that just lives on sweets, they will actually grow more slowly.
Yeah, fats especially is very helpful for the brain.
Absolutely. Remember, your brain is basically a giant lump of cholesterol, right?
It is.
I mean, obviously, I'm oversimplifying, but you get the idea, right? But the last thing a child needs is a high carb, low fat, low protein diet, right? That will make your child an idiot. I mean legitimately,
Yep.
right? And with the brain is not something that recovers, right? So you can't say, oh boy, you got really bad diet, pure sugar and carbs for the first 10 years of life. But let me switch it up now. And then your IQ
And
goes back to normal. It is not.
not only that, sir, but a lot of these products have a lot of artificial dyes and so on that are also contributing.
And the thing is, as you mentioned, that realistically, we are not really sure about the long-term implications of all of these artificial dyes. Most of these dyes haven't been used for more than 50, 60 years, right? At the most, those are the old ones, which means we do not know if there's any potential impact on stability of the genes being passed on from one generation to the next, right? We don't know any of these things. But what I can tell you is that countries that use fewer artificial dyes and things like that in their food tend to have a lower risk of things like Alzheimer's, for example. There are studies being done right now, right? So, for example, in countries like Italy and Japan that really stress a more traditional diet, heart disease, Alzheimer's, things like this are comparatively low, which is weird because Italy and Japan have two of the oldest populations on the planet. So you say, boy, they must be full of all kind of Alzheimer's, but no, but they're not. And when you check their diets are very, very low in sugars and artificial flavors and coloring. Right?
Blue zones diet, sir.
Yeah, I think that's something like that. So like you're talking about like you think pasta is pure carbohydrate, right? But guess what? The
Right.
cheese on of pasta is full of protein and good fats, right?
I know that they have been actively looking into that people who live in certain areas like Italy, for example, they tend to live longer. So they
Absolutely.
had dubbed those places the blue zones. Japan, those kind of
Right,
places
a part
because
of you used to mention France, the rural areas of France, and it's French people drink a whole lot of red wine, maybe it's red wine, that's very true.
right.
These days, it's not really wine at all. It's the food they're eating otherwise, right?
But they also live pretty active lifestyles as well.
Oh
So
yeah,
they're
yeah,
not
I'm
as
100%
concerned
correct. So what we have here is
as
a combination of factors, right? But at the
right.
end of the day, so we're still seeing where if you take somebody from the more westernized diet and you move them to one of these countries, Japan, Italy, and they start eat that diet, their health improves. So you can say their phenotype is affected once again by their environment, in this case, the food they're eating. Right. And as I said to you, particularly when they're younger, when it comes to intellectual development, the food is extremely important. Right? Anytime you see people feeding their children lots of sweets and nothing else, they're doing their children such a disservice. It's not even funny. I'd almost call it child abuse. Right?
Sure, I have a question about the Punnett square.
Yes.
All right. So whenever it is that we get something to solve, a problem to solve, right?
Yes.
Um, we should always come up with the possible gametes. That's it.
Anything to do with inheritance of genes from with of specific traits, you're
Right.
almost always going to be asked to come up with the gametes yourself.
All right. So in the PowerPoint, before we get to that, I noticed that on one of the PowerPoints, dihybrid cross
Mm-hmm.
as well. On the top, it just had the recessive traits. So
Mm-hmm.
TP, 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 TP. And on the left, it just had the dominant ones. Is there a reason why it was structured like that?
I
Or you can set it anyway?
well, remember it's the possible combinations, right? we have to look at.
Okay.
So let us say, for example, we have, hold on, I need to bring this back up.
PT, PP.
Wait, TTPP? That's like one
Yeah,
parent, right?
tall plus purple. So
That's one,
TT,
okay, that'd be
PP.
one parent.
Right. And the short parent is common T's and short and white. So TT, PP, lowercase.
Okay, so it'd be
The
like,
other thing.
so okay. So capital T, capital P for one parent?
Yeah. Yeah, no, TT, PP, yeah. Capital T, capital P, right?
Okay. And then the other parent as well.
And short and white. So common T's and common P's as well.
Okay. So here's the thing. We look at possible gametes that can come from here, right? This
Right.
one only has capital T's and capital P's to give. So
So there's
therefore,
only T and P.
well, I mean, they're all only T and P, but you have to remember the capital.
Right. Capital T, capital P. Dominant. The
Yeah, so
dominant.
this one can only produce this gamete. And on this
Okay.
side here, this one can only produce that gamete, right? So when you
Right.
set up on its square, I mean, realistically, there's no reason to do the 4. But let's say you decided to do the proper Punnett square, the hybrid one, which means, of course, a 4x4.
Right.
That always going to be a very repetitive 4x4
It is.
because you're going to have so one pair will be like that, and the other ones will be like that.
So that's what I was asking you, sir. It doesn't matter where you put the dominant or the recessive, on the side or at the top. It doesn't
No, it
matter.
doesn't. You can put it anywhere
Okay.
you feel like,
All right.
as
That's
long as you're not
fine.
mixing them up with each other.
All right.
So as long as
That's
you don't
what
have
I
some
want.
capital T's on this side and some commas up here, then that's fine, right?
Right.
But realistically,
Right.
this is unnecessary because since it's only one gamete, realistically, all you really need is something like this. Even not too much. Right? So you have,
Right.
because all the offspring will give you the same result. Right? Something like that.
Yes, sir.
All right, good. So
Yes,
remember,
sir. I have a question.
shoot. Go ahead.
All right. So remember when I asked the question about phenotype versus genotype? The reason why I asked was because I saw a group of people. I don't remember which part of the world they live, but they have the gene for like round eyes. And then because of where they live, they don't have like a lot of nighttime or the sun doesn't set. So it affects their eyes over time and their eyes they start looking Asian. So like the lid would grow over and the eyes look smaller. So I'm
All right.
wondering how is it that
Here's the
they're
thing though. The epicanthic folds you'll find in Asian eyes are not environmental. Those are strictly genetic. And they serve no, as far as we know, they don't really serve any purpose. Right? With regards to people living in places where the sun doesn't set for months at a time, you're looking at people like the Inuit. So they live up in Alaska, way up north. So yeah, they'll experience months of continuous sunshine or semi-darkness depending on the time of year. But they are the Inuits themselves, they are Asians, right? So they will tend to have more Asian features. But you will of course find them without the almond-shaped eyes sometimes as well, right? I mean, you would even find black people with
okay.
almond-shaped eyes too, because at the end of the day, pretty much because Africans are, as I say, quote unquote, the wild type of human. Basically, all the genetic traits you see in the other subcategories will show up in Africa. So you have blonde hair Africans. There's a small group of them, but there are. You have naturally blonde hair Africans. You have naturally blue-eyed Africans. You have almond eye Africans, that is natural to them. So it just so happens that it's more obvious in these other racial groupings because there's a large number of them in one place, right? So I mean, I can't think of
Okay,
any
sir, but
trait.
they were directly saying, in the video I was watching,
Uh-huh.
they were directly saying that the original set of persons, they had wider eyes, and then over time, they started to have smaller eyes, and they were saying like nowadays their children are coming with smaller eyes. So that's why I was wondering,
The children
is
are
it that?
having smaller eyes.
Yes, sir. So that's why I'm saying, is it
Smaller,
that?
not rounder.
Yes, sir, smaller. So
That
that's why I was
seems
wondering
unlikely. The
how
thing
is that
is,
possible?
there has to be something driving that. Because in order to have smaller eyes, the size of the orbital bone would have to change as well. And that's a lot of genes we're talking about. So unless there's something in the environment forcing small eyes, making smaller eyes a better choice, that seems unlikely. But if it's a small, but here's the thing. In very small populations of people, we have this thing called genetic drift, where small populations can undergo over time what would seem to us to be extreme genetic changes because of how small the populations are. Big populations have a very hard time shifting their genetics, right? That's another
Okay, sir.
possibility, right? So if it's a small tribe of people, a few dozen to a few hundred people, then you can have extreme changes from one generation to the next in a small population like that, right? When we do Hardy Weinberg, we'll talk more about it. All right.
Okay, sir.
So the idea of probability. When you look at a Punnett square, okay? The Punnett square isn't giving you what must happen. The Punnett square is giving you what is likely to happen. So for example, if we look here at our Punnett square showing the phenotype, according to this cross here, we're supposed to get one, let's see, how many wild types? 1, 2, 3, 4, 5, 6, 7, 8, 9. So we should have 9 wild types, right? And then we should also have, let me change the color of the pointer. Let's see, we have broad wings, black body. So we have one here. So one broad wing, black body. Here's another one. And here's another one. So we have 3 of those. And let me change the
Sure.
point again. Yeah.
So, sir, with the dihybrid crosses, the phenotypic ratio is always a 9:3 to 3:1.
You're first in me.
So what?
I'll go and get to it. Just give me 1 second. I will reach you. But
Sorry.
your mind is heading in the right direction. But
Oh.
right now, then we have vestigial. So short wings, striped bodies. We have 1, 2, and 3 there. And the last one, of course, we have black body with short wings. So as you rightly said, this ratio here is a 9:3 to 3:1. Okay. So what this means is that if you have 16 offspring, 9 of them you expect to look like the wild type, 3 you expect to have the broad wings and ebony body, so a mixture of the 2 traits. Another 3 should have another combination of the dominant and recessive traits, and only 1 of the 16 would be fully recessive, right? That's the basic idea. Now, this holds true from a probability perspective, right? Assuming all other factors are the same. If you're doing a regular dihybrid cross, right, then you're supposed to get this when you cross 2 heterozygotes, right? But I want you to understand that this here, 93 3:1, is a probability. It's not factuality, it's not real life. So for example, what if I ran this experiment 100 times? Okay? I ran this experiment 100 times. So each time I get 16 offspring. So I do this 100 times, I get 16 offspring off each. So that all means I have 1,600 offspring. Okay? Of that 1,600 offspring, I'm still expecting a ratio of 9, 3, 3, 1. Right? Which means I would be expecting 900 wild type, 300 broad wing, 300 ebony, well, broad wing ebony body, and vestigial wing stripe body. So 300 of those vestigial wing stripe body, and 100 fully recessive, which is vestigial wing ebony body, right? Now, that's what I would expect to get. But remember, these are probabilities. So the chance of me getting this exact number are extremely low. I might get something that looks more like this. Instead of getting 900 flat, I might get 840 here or so. Right? I might get 330 here. Then I might get 340 here or so. And I might get 90 there or so. Does that add up? I can't even tell. Let me see. Yeah, I think that adds up to 1,600, right? So I still have 1,600 offspring here, right? This is what I would have hoped to get up here, so. There you go. Right? Based on the ratio, that's what I would have expected to get, right? So 9,331 ratio, I would expect to get this distribution if I had 1,600 of these flies, okay? But instead, I get this distribution. Why? Because probability is not certainty. So when I say, so I'm saying 9 times out of 16, I should get a wild type. 3 times out of 16, I should get a broad wing. And 3 times out of 16, I should get a vestigial, right? So these are chances, right? Y'all hear lotto. So y'all have an idea of what we want to talk about chances, probabilities of getting things, okay? Now, sometimes you have to be able to look at 2 sets of numbers and ask yourself the question. This here, 840, 330, 340, 90, is not exactly like 900, 300, 300, 100. But then we have to ask ourselves, is it close enough that we can say, sure, it's good enough, right? That is now when we get into the statistics of the chi-square tests, which we'll be doing later as well, right? So the Punnett square gives us the chances of something happening, the probability of it happening. But our numbers are rarely ever going to be exactly like the proportions we expect to get. We have to determine if the numbers we get are good enough that we can accept them, okay? So keep that in mind, right? So chi-square equals probability, because we never know for sure what we're going to get, but that's what we expect to get. It's the same thing I was saying, boy. What's a okay? Here's a kind of a messed up probability. But if I say to you, right, and you can tell me if this sounds reasonable. According to US Census, US data, right? So this is their data. You are 5 times more likely to get robbed by a black person than a white person, right? So you're 5 times more likely to get robbed by a black person than a white person. Does that mean you have 5 times more black robbers than white robbers? No, it doesn't mean that. Right? It might simply mean that you have a few black robbers that are very, very good at their jobs. Now make everybody else look bad. Right? So you have to be really careful with statistics and how you interpret them. Okay. But I think you all did a statistics course already, so you should have some idea about that, right? Just like say, boy, they say the chance of you getting shot. You know that Jamaica is the quote unquote murder capital of the world, right? I don't know if you've heard that term before. Y'all know that. I don't know if you heard that. I think it's what 2023, we were the murder capital of the world. Which means that statistically speaking, your chances of getting murdered are higher in Jamaica than any other country in the world. Right? But let me ask you, any of you are really worried about getting randomly murdered on the street? I mean, it's not really something you worry about, is it? Why? Because for the most part, you stay out of the areas where people are getting randomly murdered. And it's not normally random in Jamaica, it's usually some argument between idiots or gang war or something like that, right? But even with that, I can say, don't go to Jamaica. You're more likely to get murdered in Jamaica than anywhere else. And statistically, it is correct for me to say that. But the reality of things, of course, is different. As long as I am not within certain groups of people and in certain places in Jamaica, I am, for the most part, perfectly safe. Right? So you have so many tourists come to Jamaica and say, oh, they don't want to leave the hotel. They might get murdered or raped or whatever, right? And they are, for the most part, perfectly safe, right? Anyway, so that's statistics for you, right? So remember, understand probability. Probability is a chance of something happening. It's more likely to happen than not. All right. So the classic, of course, is the coin flip. Now, I'm assuming you guys would have done like coin flipping in high school, the experiment, where you're given coins, and you're told, coins have a head and a tail, and you flip the coins, and the idea is that there's a 50-50 chance that when you flip a coin, it will land on the head or the tail, which is why, of course, people, I don't know if anybody still does this, you're trying to make a decision, you're not sure what. You have two possible outcomes. You just flip a coin in the air, grab the coin, and look which side comes up, and use that to base your decision on, right? So the idea is, of course, you're putting it in the hand of the universe. Let the universe decide because I don't want to. Right? Now, is the probability actually 50-50? Actually, no. It skews slightly towards heads, weirdly enough, right? Very slightly, but it still skews towards heads. Anyway, so now hopefully you understand the whole idea of probability versus reality. Probability, the chances of something happening, the greater the chance, the more likely it is to happen. A certainty, something did happen. Okay? Okay, so sources of genetic variation. We already spoke about independent assortment and how the genes are put into the chromosomes. What am I saying? How the chromosomes are put into your gametes randomly, right? You never know which one of the two pairs of alleles will be shoved into which gamete. But we also have, of course, mutations. I'm assuming you are at least somewhat aware of the idea of a mutation, right? Unfortunately, the Marvel movies have done a real disservice to the idea of a mutation. Because what they have in the Marvel movies ain't no mutation, right? You can't wake up one morning and can't fly. That does not happen in nature. Mutations are, of course, though, spontaneous changes to your DNA. These changes usually happen before birth, though. Okay? Spontaneous changes to the DNA. What do I mean by spontaneous? I mean, it just happens out of the blue during the process of putting the gametes together, right? Mistakes are made, and then the genes are improperly coded, for example. So when they're passed on, the genes are coding for something different or than what they originally started at. So when now that offspring is put together, they are put together with genes that don't do what they should do. Some cases it might be a benefit. Some cases it might not be. For the most part, most mutations are actually what we call neutral, right? So they're not bad, they're not good, they're just there. For example, what is the genetic advantage of having brown hair versus black hair? I mean, there isn't any, right? Or blonde hair versus, there might actually be a disadvantage of having blonde hair if you live in a very hot place, right? But so most mutations are just they're not that important. I can say the same thing for what you call it there, eye color. Even though blue eyes are slightly disadvantageous, people with blue eyes tend to lose their eyesight more as they get older, right? But because it's as they get older, they would already have had their children anyway, so it doesn't really matter to genes, right? So for example, you have people who are born with what do you call it again? Uh, double joint. You can bend your finger over in several different directions. I mean, there are no real benefits to a mutation like that. Unless you're picking fruit or something, maybe, I don't know. So most of mutations are going to not be negative, just be neutral. You know? Though some mutations will occur that can actually pass on a significant benefits to people, right? And if the mutations are good enough, then evolution can work on these mutations and ensure that these mutations get passed on more often to the next generation. And over time, the mutations spread in the population and become the norm, right? And that is essentially how evolution works. Evolution must have mutations in order to function, okay? We will, of course, be looking at evolution later on, but just keep that in mind. You need to have changes, differences within a population for evolution to work. Mutations are a good way of producing differences, okay? All right, we already did law of independent assortment already, right? So, but as it says here, you can mathematically predict possible combinations by using a simple formula. So you have number of possible genotypes is 2n, where n is the number of genes or traits to be considered. All right. So if you have 100 traits looking at, then the possible combinations are 2 to the 100th power. Right? And that equals 1.26765 times 10 to the 30. Right? That's billions, hundreds of billions to 1. So if you're just contemplating the chances of 100 given traits showing up in 2 people, the exact same 100 traits, it's billions to 1, much less, okay? So yeah, chance of somebody looking exactly like you, having exactly your genes, and you're not maternal twins is extremely unlikely. All right, I think we went through these terms already, so we should be good for those. We did this already, and we did that already too. So all this should be good. And right, we did Punnett square, and we did dihybrid cross a while ago. So all should be well where that is concerned. I feel like I'm going backwards. Okay. Now, let's look at some human hereditary conditions, right? I'm going to look at 2 of them. I'm going to look at cystic fibrosis, and then I'm going to look at sickle cell anemia. Here's the reason why. Cystic fibrosis is the most common genetic condition among Caucasians, right? And of course, sickle cell anemia is the most common amongst people of African descent. All right. So that's the reason why we normally choose those two. So cystic fibrosis. Generally speaking, what cystic fibrosis does is it causes all right, how do I simplify? Let me simplify. Wait, sorry. Alexi having Wi-Fi issues. Okay. All right, no problem. I've noted. Okay. So cystic fibrosis is a serious issue, okay? It causes the mucus produced by a person to get very thick because of an issue with chloride atom transport. But I'm not gonna get too deeply into it, right? But it's a chloride transport issue. That's what's important. And because of that, the mucus gets very thick and it gets worse over time. Now, that might not sound like that big a deal. Oh, okay, mucus get thick. Little hard as a cough or cold, right? What's the big deal? We always forget that mucus is everywhere in our bodies, right? It's in our joints. We produce it lining our lungs. We produce it within our lung tissue, nasal cavities, right? There's mucus all about the place in human beings. Digestive tract, there's a lot of mucus going on in there. Now, if your mucus is extra thick, it makes it very hard to move, right? So you can imagine over time, mucus starts building up, for example, in your lungs, and it is so thick and heavy that you can't just cough it out, you can't clear it out. So over time, the mucus builds and starts filling your lung alveoli, and there's nothing you can do about it. And you slowly, essentially drown in your own mucus because you can't do anything about it, right? And of course, while that is happening, your digestive system starts giving problems because the thick mucus starts blocking up parts of the digestive tract, right? So it's hard for you to pass food through the tract, right? And all sorts of things. So cystic fibrosis is not nice, right? Usually it kills the people who have it, right? But it normally kills them later in life. Now, what you'll find with genetic diseases is that the ones that kill you quick are usually not the problem. It's the ones that take a little while to kill you that are the problem. Because if it kills you quick, chances are you're not going to have children, so you won't pass on the problem to somebody else. But the ones that kill you slowly, those are the real horrible ones, right? Now, as I said, it affects 1 in 4 children are carrying the gene for cystic fibrosis. As it says, CF disease affects transporting tissues. Mucus is accumulated in lungs, cause infections, and if you don't get infections, as I said, your lungs slowly stop functioning until you die. Okay? So if we have 2 parents carrying the recessive gene for cystic fibrosis, which of course is recessive, it's common C, right? So we can see here we have 2 parents that are heterozygous for the trait here. So we have 2 heterozygous going. So when you cross now, because remember, if both parents are heterozygous for the cystic fibrosis gene, you have 1 big C as a possibility, 1 small c for both. Now look at our ratio. We have look at the genotypic ratio first. We have 1 big C, which realistically is the one you want to be, because this one has no trace at all of cystic fibrosis. You have 2 small c's, right? So these 2. And you have 1 now that has the cystic fibrosis. So you have a genotypic ratio of 1 to 2 to 1, which is the normal genotypic ratio you expect when you're crossing, when you're doing a standard monohybrid cross with dominant and recessive. So according to this here, right, you have a 25% chance of having a child with cystic fibrosis. Because I can say 25% here, 50% here, another 25% out here. And those, of course, add up to 100%, right? Now, remember again, this is probability, this is chance of something happening. So what does that mean? It doesn't mean if you have 4 children, one of them must have cystic fibrosis. It means that you have a 1 in 4 chance every single time of having a child with cystic fibrosis. You could have 4 children and all of them come out 100% normal. That is possible. You could also have 4 children and all of them come out with cystic fibrosis. That is possible, right? Both of those scenarios have the same probability of occurring, right? But based on what we have here, chances are you have a 50% chance that the child will come out and be a carrier. So in the next generation, the percentage of carriers probably will not go down significantly, right? Now, if you cannot reduce the number of people who are carrying the gene in the population, you're not going to be able to reduce the presence of the gene in your population, right? If tomorrow they said, you know what, we're going to just genetically screen every single person, every single white person, and if you have the gene for cystic fibrosis, sterilize you, no children for you, right? Guess what? Next generation, no one will have cystic fibrosis, right? Because you would have removed the ability of the gene to be passed on to the next generation. I mean, I don't see that happening. You know, there are some human rights implications to that, I'm sure. You can think of them. But realistically, if you want to stop a genetic disease, that is what you're going to have to do. You're going to have to go in and say, guess what? Everybody with a genetic, with even one of the bad genes, no children for you. Now, you can imagine the likelihood of people saying, oh, all right, no problem. I guess that's okay, right? That's going to be highly unlikely. Now, that said, you can do DNA screening, you and your partner, but not us, we're not white, to determine if you have cystic fibrosis present. But by the way, I will say this. Even though I started out saying it's a white people disease, please understand that the reason why this disease was mainly in Caucasians was that before there was a lot of racial mixing, of course, they would have been passing that disorder amongst themselves. That disorder would have been a mutation occurring within their population. But it would not have occurred in the black population in Africa or the Indian population or anybody else. So that mutation would have been unique to Caucasians at the time. But remember now, once you start intermixing, it means, of course, eventually you're going to start seeing cystic fibrosis in people who are not technically considered to be white people, right? It's only a matter of time. But I mean, can I say this to you? That from a scientific perspective, race makes no sense at all, right? Racial groups make no sense. All a racial group is a isolated community with certain genes, specific genes. They have more likely to be in common than others. That's all, right? If you leave the world to allow intermixing, in a few generations, there won't be any white people, there won't be any black people. Everybody will look vaguely Asian, if I'm honest. Because, you know, most of the world's population are Asians, right? So if we did that, come back in 500 years, everybody just looks kind of Indianish. Or sort of dark Chinese. And that would be the entire world, right? Because human beings really don't have races from an evolutionary perspective, right? Anyway. Now, here's another interesting disease. And then I'm going to do sickle cell. So gotcha disease is a rare genetic disease. It causes a lipid storage disorder. So what basically happens is that fat start to accumulate around your spleen and your liver and your bone marrow. Now, bone marrow is a serious issue because, as you know, that's where blood comes from, right? But notice it is very common amongst people of Jewish descent, Eastern European Jews, right? Why is that the case? The Jews are an interesting test case, okay? If I'm honest, most people will have a hard time defining what a Jew is. For most people, Jews live in Israel, right? But most of those Jews that you see in Israel now are actually from Europe. They're of European descent. All right. They're more cultural Jews than genetic Jews. Most of them don't really have any genetic link to that area of the world anymore. Okay. Their ancestors would have left a very long time ago. So there tend to be, so genetically speaking, Jews are a mixture of Eastern European Caucasians as well as the equivalent of people from Turkey, right? So it's sort of half Middle Eastern, half European kind of people. But here's the thing. Because of their religious identity, they tended not to mix with other people. Sorry, give me one quick second, please. Hello? Yes. Eight and 12. Yes, I should be available. All right, thank you very much. All right, bye-bye. All right, sorry about that. All right, so see if this makes sense to you. When you have small groups of people living in a country, right? But they have separated themselves from the wider country. By that I mean they do not interbreed with the rest of the population for whatever reason, okay? In the case of Jews, of course, it's a religious thing, right? I think most people know that Jews believe themselves to be what they call the people of the book, right? They're God's chosen. And again, lots of religions believe something of that nature, right? But as God's chosen people, they might talk to somebody who's not a Jew. They might go out and have a drink with you, but they're not going to marry your daughter, right? So what you tend to have happening is that you have a lot of Jewish communities that is inbreeding is a real issue, okay? So now you have a relatively small population of people that are inbreeding. So it's very easy to take a bad gene and spread it within that small population quickly, right? So in the case of Gaucher's disease, that's what happened. So this, of course, speaks to the importance of outbreeding to maintain what we call vigor, right? If you have a small population of cousin and cousin going on, it never turns out well. Okay? All right. So as it says, the incidence in the Eastern European Jewish population is 1 in 500, but it's 1 in 100,000 in the rest of your European population. So remember, the Eastern European Jews have been in Europe for over 1,000 years, and they have an incidence of 1 in 500. Everybody else, 1 in 100,000. Why? Interbreeding of basically cousins, right? So, in Jamaica, when I was a cousin and cousin boil good soup, not really, right? Now, let's talk about sickle cell. All right, the sickle cell trait, when I'm going to write it this way, HB, that's for hemoglobin, And then we have a superscript S. Okay? So HBS equals sickle cell, okay? Whereas HBA, like that, equals normal, right? Now, the reason we write the HB and superscript S is because sickle cell is a bit strange. It is not Mendelian. Sickle cell is not dominant and recessive. It is what we call codominant. Right? It's codominant. By that I mean, if both genes are present in a heterozygous, both of them are trying to show up in the phenotype, okay? So let's say you have somebody who has a genetic combination like this, HBA, HBS, okay? We call this person a carrier, okay? About 7% of the population in Jamaica carries the sickle cell trait, okay? So realistically, at least one of you in here probably carrying a sickle cell trait right now. Of course, if you're HBA, HBA, you're going to be normal. Okay? And if you are HBS, HBS, you're going to be, as we say in Jamaica, a sickler. Okay? Now, that of course means that we can do a Punnett squared to determine the chance of you being either a sickler, normal, or a carrier. Okay? Using our regular Punnett square method. And then I'll talk about what are the ramifications of being a carrier versus normal, etc., right? So let's say we have two parents that are HBA, HBS, okay? So they're both of them are heterozygous for the trait, okay? Now remember, that's 7% of the population, right? So the chance of two people with the sickle cell trait meeting and having a child will be less than 3%. Okay? But 3% of 3 million is a whole lot of people. Right? 1% of 3 million is still 30,000 people. Okay? So we're not talking about an insignificant number of people. So let's say what happens. So no, remember now, they have two genes that they can pass on. Either the HBA gene or the HBS gene, right? Let's get rid of this. So remember, codominant. So HBA, same thing up here. Now, because they are codominant, there's no recessives, right? They're codominant because they're both contributing to the phenotype, okay? So please keep that in mind. So the first offspring here, we have HBA, HBA, just perfectly normal, which in Jamaica, that's what you really want. Then we have HBA, HBS. Realistically, you could always write the S first, but you'd have to, but I started with A, so I keep going A, right? So remember, whichever one you start with, that's what you have to keep going with. Next one here, HBA, HBS again. And the last one is, of course, HBS, HBA. S, okay? So the genotypic ratio is 1 normal to two carriers to 1 sickler, okay? Well, what I should say is that 1 HbA, which of course phenotype makes you fully normal. Then we have 2 HbA, which means you carry the trait. And you have 1 HbS, which means you have full-blown sickle cell, okay? Now, if I said phenotypic ratio here, normally I would expect you to say, oh, 3 to 1, okay? That's what you'd expect. But remember, this is codominant. Because it's codominant, it means that as long as the gene is present, it is going to cause some effect. All right, the bad gene. All right, I'm going to explain why the term bad gene might not necessarily apply, okay? It's a little bit more complicated than that. I don't want to just say, oh, bad gene. But here's the thing. The phenotypic ratio is going to still be 1 to 2 to 1, okay? Because I have 1 fully normal, then I have 2 codominants, and then I have 1 full sickler, right? Now, let me explain why. The 1 fully normal, of course, they have no sickle cell trait at all. Their hemoglobin is all standard hemoglobin. As you know, sickle cell affects the hemoglobin. Their blood cells, remember in sickle cell people that the blood can be different shapes, red blood cells, including sickle cell shaped. So if you're fully HbA, you're 100% normal. You look at your blood, it's perfectly fine, okay? If you carry the sickle cell trait, you are not 100%. If you have the sickle cell trait, some of your blood cells will carry regular old hemoglobin. So those cells will be perfectly normal, but some of them are going to carry the sickle cell hemoglobin, right? And that sickle cell hemoglobin is what alters the shape of the cells to give the red blood cells funny shapes. So if you're codominant, you have some good red blood cells and some sickler cells in your body, okay? Call it 50-50. If you are, on the other hand, a full-blown sickler, as we say in Jamaica, all of your blood cells are going to have the abnormal hemoglobin in them. All of your blood cells are therefore going to be distorted. The shapes are going to be bad, right? Once you have these very distorted, very bad shaped cells, they get stuck in blood vessels all the time, right? Because of how badly shaped they are. They have a very, very hard time passing through narrow capillaries. They bundle up in the narrow capillaries and cause problems. Also, they hold less hemoglobin because of the misshapenness. So they're more likely to cause things like clots and blockages. They also carry less hemoglobin, which means they carry less oxygen, which is why we call it the condition sickle cell anemia. Because if your blood is carrying less oxygen, you are going to be anemic, okay? Now, in terms of bad or good gene, obviously, having sickle cell disease is bad, okay? But what about if you are codominant? 7% of Jamaicans. Having those sickle cells in your body does have a negative effect, okay? And what I'm about to tell you is generally, okay? So generally, people with the sickle cell trait, as we say in Jamaica, have an increased risk of anemia, for one, right? So that's one definite disadvantage. They also tend to have smaller lungs, right? They also tend to have a harder time building muscle because one aspect of building muscle is providing the muscle with oxygen to feed it. So if you have anemia, it's harder to feed the muscles, right? So they also have a harder time putting on positive weight. Like once again, muscle weight. So they have a tendency either to be very thin with smaller lungs and over a weaker body, or these days because of the diet, they tend to be heavier, but once again, physically weak. Okay? So those are the disadvantages of having a sickle cell trait. That said, if you happen to live somewhere where malaria is a real problem, right? That malaria is a killer. Malaria to this very day is still one of the biggest killers of people all over the world, right? If you happen to live in a place where with lots and lots of malaria, you will find that having the sickle cell trait is a positive because the malaria parasite has a much harder time infecting a sickle cell. So you get bitten by mosquitoes countless numbers of times. If you catch malaria, you might get sick, but chances are that you will not die. You will live long enough to have children of your own and pass on your genes. If you are fully normal HbA, you have a more than 50% chance of dying from malaria. Right? So yes, you're stronger, you're faster, whatever else, but mosquito kill you. Right? So in terms of the sickle cell trait being good or bad, it is situational. It is heavily dependent on the presence of malaria. If there's no malaria, then definitely sickle cell trait is not good for you. You don't want it. There's no malaria in Jamaica, so there's no real positive to it. At least there is some malaria, but the probability of you catching malaria is fairly low. Okay? So yeah, that is why sickle cell is a problem in Jamaica, right? All right. Any questions here, or can we move on? All right. Now, if you do happen to have a sickle cell trait, I would advise that when you have decided to pass on your DNA to the next generation, that your chosen partner, you both get DNA tests, right? To verify whether or not you all both have the sickle cell trait. Because remember, if you have the trait, you have a 50% chance of having a child that also contains the trait, which does not really help the overall population. And you have a 25% chance of having a child with sickle cell, which of course in itself would be a problem. Okay? And if you've never ever known anybody with full sickle cell disease, without treatment, most of them do not live to the age of 12 without treatment. Right? Even with treatment, they average out in the 50s. So they tend not to live very long lives. Right? So yeah. If you have a trait though, of course, it does not really affect your longevity. So keep that in mind as well, right? All right. We've done dihybrid cross already. We don't need to do it again. This is just dihybrid cross talking about colors of flowers and length of stem. So we can leave that one alone for now, right? We've already done it. We already spoke about independent assortment already. Okay, test cross. We haven't done test cross yet. A test cross is a way of potentially determining the presence of a recessive gene in a organism of unknown genotype, right? Normally, these things are used in horticulture. Horticulture, flowering plants usually, right? Horticulture different from agriculture, right? Now here's the thing. Human beings are kind of funny. There are certain things that we just like without any logical reasoning behind it. For example, lots of us like pink flowers. There is no evolutionary benefit to enjoying the pink flowers or anything like that. You just see pink flowers like, oh, that's so pretty. I want it. Right? And of course, that means that you're willing to pay money for it. So having an ability to determine a color that will corner in the market and in producing flowers that only produce that color you want is very important. Okay? So that is where we come now to our test cross. So let's say, for example, for whatever reason, pink, as I said, white flowers are the rage, let's say, right? You want white flowers for whatever reason, right? You have, white is recessive, usually, remember, right? So white, what's that like that? So white, of course, is recessive, usually. And let's say the red flower is dominant because it normally is, right? By the way, something interesting you probably know. A lot of flowers that are white, right, are actually only white to the human eye. I don't know if you ever heard this before, but sometimes you'll see bees being attracted to white flowers. They're like, why are they attracted to that weird white flower? Because some flowers, they reflect light in the ultraviolet spectrum, right? And in the infrared spectrum. Human beings, our eyes cannot see infrared or ultraviolet light. So of course, when we look at the flower like that, it tends to default to us as white, right? So when the bees are looking at the flowers, they're seeing all kind of psychedelic colors that look crazy to them. So they're drawn right to the flower. You look at it, it just look like a sheet of paper, like, what's the big deal? You simply cannot perceive the colors that the bees are perceiving. Okay? So that's the reason why you will have a lot of successful white flowers. Because to the eyes of the insects, those flowers are not white. Like trumpet flowers, for example, right? Anyway. So we have somebody who just want white flowers, right? So they want to grow exclusively white flowers. They have to find a way of identifying flowers that have the white gene in them so that they can breed them together to potentially make white flowers. Okay. So here's what you do. If you're given a red flower, and of course red can be either homozygous for red or heterozygous for red. So it has a recessive gene in there. You need to know what this red flower is. See if it's worth the trouble of crossing with another red to get white, okay? What you do is this. You find a recessive flower. In this case, you find a white one. All right? And you cross the flower of unknown genotype with the one of known genotype. Okay? And what you have happening is this. You're going to get particular ratios. If you cross a fully homozygous dominant with a fully homozygous recessive, I'm sure everybody remembers you're going to get all heterozygous dominant. So that means all of these flowers will come out red. Okay? This red. So you know that red flower is of no use to you. But if you cross those flowers and instead you get half of them being red, half being white, it is because you have this combination. right? So 2 white flowers, 2 red flowers. So by doing a test cross, you can identify flowering plants. Well, for a specific gene, you can identify whether or not they're heterozygous or homozygous for that gene using the test cross. Okay. So that is the purpose of the test cross, to determine the genetics of an unknown, usually plant. Okay. And you can also do test crosses in dihybrid plants. But remember, dihybrids, you're looking at 2 different traits. So it means you have to cross them with a plant that is double recessive. Okay. So your test plant has to have 2 recessive traits for the 2 traits you're trying to test out. So in this case here, we have a tall plant with purple flowers. We want to know if they're heterozygous for these traits. So we're crossing it with a short plant with pink flowers, since pink is recessive here, right? So what they're showing here is the possible combinations that can give you tall and purple, okay? So all of these can give you tall and purple. So this plant could be any one of these. Whereas over here, you know what your recessive one is already. So you have one known versus one unknown. So when you cross them now, depending on the ratio you get between white and tall and purple and short, you can determine which one of these that plant here belongs to. Okay? All right. Hopefully that makes sense to you. We're coming up to the break. But let me just do a couple more slides before that. So we're about to start the exceptions to Mendelian genetics. So modifications or additions to Mendel. Remember, Mendel was about dominant and recessive. Mendel did mention things like codominance, which is what I mentioned when we were talking about sickle cell. He also did mention things like incomplete dominance, which we need to cover as well, okay? So for example here, we have red flower, pink, and white, okay? So how do we get pink when we have 2 genes? We have gene for red flower, gene for white flower, we end up with a pink flower, right? So how does that happen, right? How does a red horse and a white horse give you a brown horse? That sounds crazy, right? So we're gonna talk now about that aspect of well, it's basically non-Mendelian genetics, and try to explain how that works out. All right. Let's take 15 minutes here, please, and then come back. Let me see what time I have here. All right. One second here. I need to stop the show. All right. I have 7:06 here. So let's come back at 7:20 now. And then we can continue from there. All right. So see you all in 15 minutes or so.